William Kelton. We're calling him Will because we've got another William on the pillar lead. So he's looking after target design. And Will originally did his degree at University of Canterbury, chemical engineering degree, and went to Austin, Texas to do his PhD. Made his way back to NZ via Scion and Aura Therapeutics, and now is a senior lecturer at University of Waikato. And he's going to talk to us today about enzymes for site-specific modification of RNA. Thanks, Will.
Excellent. All right, kia ora koutou, and thanks very much. It's nice to see you all here today. So, as Gary mentioned, I'm from the University of Waikato. For some of our international visitors, that's just slightly south of Auckland by about an hour. And if you're into concrete 70s architecture, this is the university for you. So I'd like to upfront thank some of the team that I've been working with. So one of the cool things about setting up this platform has been the opportunity to work in pretty interdisciplinary teams. And so I'm just going to take you through a project today that sort of started before the platform and then is sort of ongoing and some of the ideas and where we think we might be heading with it. So I'd like to acknowledge, of course, at the University of Waikato, a couple of my collaborators, Dr. Joanna Hicks and Adele Williamson, who is here as well as a PhD student who's just starting. And we've been working and are indebted to the folks down at Victoria University of Wellington, Lisa Connor, who you will have seen many times today, and Professor Wayne Patrick, who is here as well as one of their wonderful PhD students. We're rolling in a few extra collaborators to this sort of space as we go along and setting things up. Down at the University of Otago, Associate Professor Paul Gardner is a computational expert, and we use some of his expertise, and then also talking to a few people overseas. Oh, it's gone. Excellent. Right. So, we've been thinking a little bit about modifications of RNA, and as part of the payload pillar, looking at ways that we can bring maybe some new technologies into New Zealand to support the efforts of manufacturing in the long run. So, we've long known about mRNA modifications. Of course, people will be very familiar with the pseudouridine or derivatives thereof. But there's been a lot of other modifications, and we've known about these for a very long time. So you can go all the way back to 2005, and some work from arguably the people who are pioneering in this field, so from the Kariko lab, where they had a look at various modifications on RNA and its ability to elicit immune responses. Now, of course, pseudouridine and some of those species are really good at reducing immunogenicity of these constructs. That's really good. You still might want a little bit to generate good responses, but there's some instances where you definitely don't want as much immunogenicity. And so one of the lesser studied modifications is this M6A modification that I'll talk to you about today. Now, I'm going to sort of tell you the story in the context of circular RNA, which is a technology that's emerged in the last couple of years. And it's a technology that's had some challenges, as many of you will be aware. So making some of these circular RNAs is challenging, but only in the last maybe 12 months there's been a number of groups, particularly from the US and some groups out of China, who have started to solve some of these problems using approaches like self-splicing introns to make these constructs. And they're really massively improving the efficiency by which you can make these constructs. Now, one of the challenges is it's difficult to modify these circular RNA constructs. Things like pseudouridine mean that the RNA doesn't form or doesn't assemble very well in circular format. But if we turn back to nature and have a look, circular RNAs are actually quite common in a number of our tissues. Particularly, I believe, our brain tissue, we have a large number of circular RNAs that are expressed there. And a number of these circular RNAs contain this methyladenosine modification. And it's really important for controlling not only the expression of genes from the mRNA, but it also has a role in how inflammatory these RNAs behave when you put them in vivo. And so what Lisa and her team investigated was to take their SARS-CoV-2 RBD vaccine and have a look at it in circular RNA format, but with various levels of these M6A modifications doped in. This is just during the synthesis of these. And so they've run a whole number of mouse experiments, and I'll just show you a couple of these, thanks to Abby, who collated all of this work. And so once these mice are immunized, and then 6 hours later some sera is taken, and we're looking at cytokines in the blood in this particular instance. And what you can see in blue here is when you don't have any modification on your circular RNA, you're getting these really strong inflammatory responses. If you use pseudo-methyluridine, then this is in a linear construct, that massively reduces some of your inflammatory markers. But surprisingly, or strikingly I should say, when you have even 1% modification of these M6A nucleotides in there, you're getting a massive reduction in these inflammatory responses. And this is across a wide range of cytokines that Lisa and her team looked at. So they went a little bit further and had a look at some of the innate cell populations. This is after 24 hours, isolating lymph nodes, having a look at conventional dendritic cells, both the frequency and the total number of these cells. And again, these modifications are massively reducing the levels of these cells, so suggesting that the innate immune response is not activated as strongly. They also had a look at some other markers of dendritic cell maturation, like CD86, and also some of the interferon response markers. Again, just showing that when you include these modifications, you're massively dampening the inflammatory response. So together, the team that we've put together thought this was pretty neat and something that we might like to explore a little bit further. And there's a couple of questions that we have about this. So, for example, we're down at 1%. Can we modify the RNA less in order to see the same kind of phenotype? And then also, at the moment, it's a very non-specific process. Is it also possible to kind of target some of these modifications to particular regions of RNA, especially given in circular constructs where you modify matters, especially if you're using like an internal ribosome entry site? And so some of the expertise at the University of Waikato is in enzymes and the design of these. And so I'll just take you through a few of the ideas. We don't have a lot of data on some of these at this point, but a few systems we're thinking to kind of explore this in a little bit more depth. Okay, so the first of these, which was talked about earlier today, is using some of these new CRISPR systems, in particular CRISPR-13A. But instead of using an active nuclease, you can instead make mutations to the protein that remove the nuclease activity, and then this enables you to use it as a pure targeting kind of molecule driven by the sequence of your guide RNA here. And there's been some work in the last few years fusing the dead Cas13A protein to some of these methyltransferase enzymes, which are able to create these modifications. And so the idea in this particular case is that you'll be able to target guide RNAs to try and modify some of these circular RNAs to specific regions to see what their effects might be. Now, this is straight from the publication, not our data, but when they tested some of these constructs and a couple of different methyltransferases, they were able to see a couple of orders of magnitude upregulation and site-specific methylation when they were testing these particular protein fusions. Another approach that we've been in discussion with a collaborator from the University of Western Australia, who works quite closely with Archer Fox, is Charlie Bond. And his group has been responsible for developing a series of pentatricopeptide repeat proteins. And so for those of you who are familiar with sort of zinc fingers and the tail technology, they're a similar type approach, but designed for working with RNA. And so they're a helix-turn-helix protein that are specific for an individual RNA base. And when you join these together, you can create a concatemer of these units that will have specific sequence recognition of particular stretch of RNA bases. And so the idea being here, you can join a number of these repeat units here together, add some capping proteins to improve the stability, and again, just like with the CRISPR-Cas9 system, add some sort of effector, in this case a methyltransferase, in order to make those M6A modifications. So we've had a few conversations with Charlie in order to set up the design of some of these particular proteins. And finally, an area that there's actually quite a lot of active research at the University of Waikato, both in Dale Williamson's group and Joanna Hicks's group, is to have a look at RNA ligases and how these could potentially be used for applications within the platform as well. So Wayne Patrick and Joanne Hicks recently had a nice paper in Nucleic Acids Research where they had a look at the ability of some thermophilic RNA ligases to join pieces of RNA together in vitro. And I'm just showing you one of their RNA ligases here that they isolated. a wild type form here, so we're looking at a shift up in the gel here, so higher indicates that it's ligating RNA together. And they're able to also do some engineering and find mutations that would increase the efficiency by which you could join some of these pieces of RNA together. So just providing options for before you do your in vitro, or after you've done your in vitro translation, just ways that we might be able to manipulate or modify the RNA that's being produced. The other interesting thing is a number of these RNA ligases can also work with xenonucleic acids or modified bases. And so one of the hypotheses is some of this work could also flow through into modification of linear RNAs. And this is some work taken from Adele Williamson's group as well. And so she's really interested in using some of these special base modifications, particularly in order to try and improve stability properties, looking at things like that poly A tail. But there's a number of questions here around which kind of backbones might provide best stability, also how we might scale some of these things up, and also what the impact of some of these bases might be on translation, which is also incredibly important. And so just to show you a small amount of data here, so this is working with DNA, but the same principles apply to RNA with some of the other ligases that are under development. So she's been creating a library with a diversity of these unnatural bases, and then exploring the ability of these ligases in order to join or splint together these nucleic acid strands. So again, looking at these sort of gel shift assay type approaches. And so they've found a couple of interesting ligases, which I'm just showing you down here, that seem to have quite a wide ability to tolerate some of these unnatural or xeno bases in order to splice and join these nucleic acids together. So pretty cool technology that we're keen to explore in a little bit more depth. Oh, and just some of the unnatural base pairings there. So just sort of final thing to finish up, so we make sure we're nice and on time. The other sort of thing that we've recently acquired at the University of Waikato is some small-scale kind of pilot fermentation capacity, which we're going to work with the central platform in order to investigate the production of some enzymes, like some T7 variants for them as well. So just be aware, it's sort of a 4 by 1 litre parallel bioreactor system, so if there are people who are interested in doing small-scale ferments, if you've had enough of shake flasks, I feel your pain, you're welcome to come and have a talk to us about that as well. And with that, I'll thank you very much, and I'm happy to take any questions.